Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Well, welcome everybody again to another episode of our show. Today, bringing you another really excellent guest involved in creating a better tomorrow for many people out there. Today, we have the honor of being joined by Mike Kelly, who is President and Chief Executive Officer of NervGen Pharma, which is a clinical stage biotech company. They're dedicated to developing innovative treatments to ultimately enable nervous system repair in the settings of traumatic injury and disease. And their lead drug candidate, known as NVG-291, is currently being evaluated in phase 1b/2a clinical trials, an initial target indication, a spinal cord injury. They've also initiated preclinical evaluation of another development candidate, NVG-300, in areas of ischemic stroke, ALS, as well as SCI. Mr. Kelly brings over 3 decades of pharmaceutical experience to the company, playing really instrumental roles in the creation, development, and strengthening of several companies. Most recently served as president of the United States operations for Adapt Pharma, played a major role in developing and commercializing NARCAN nasal spray in the United States and Canada, and their eventual sale to Emergent BioSolutions. Prior to that tenure, he served as the CEO and director of Covis Pharmaceuticals, was also a member of the founding management team of Azure Pharma, which later was merged with Jazz Pharmaceuticals, where he went on to be senior vice president of sales, several other roles at Guilford, Viro Pharma, TAP. He did his background, a bachelor's degree in business administration from College of New Jersey, went on to do his MBA at Rider University, and we're honored to have him with us. So a lot to get into today, a really important set of topics. Mike Kelly, thanks so much for taking time to come on the show.
Thanks, Ira. Appreciate the opportunity.
Yeah, no, it's great having you. Before we get into all things nervous system and regenerative medicine oriented, would love to hand things over to you just for a couple minutes to talk a little bit more about you. You know, I mentioned that you educated yourself here in Jersey and went sort of fast and furious into the pharmaceutical industry. Tell us a little bit about your background. I mean, I know you spent a lot of time on the commercial side, but what interested you in, aside from being in Jersey, the sort of the epicenter of the global pharma industry, in getting into this space in the first place?
Yeah, actually, Jersey had a lot to do with it. Actually, my father worked in accounting, and he worked in a lot of the pharmaceutical companies up and down Route 1. So I think, he used to come home with boxes of J&J products from the J&J store. And, we talked a lot about pharmaceuticals and pharmaceutical companies in the household when I was growing up. But several, I guess, I'm the youngest of 5, and several of my siblings had friends and or boyfriends or relatives that had gone into the pharmaceutical industry. So during a lot of the family gatherings and talking about what people did and where they worked, I was always kind of intrigued with pharmaceuticals. And right out of college, just like every other kid, I tried to get in and I couldn't. It was like that Army commercial, the door slams in your face and get experience, kid. Come back. But I went away, I got sales experience, and then ultimately got my break and got into TAP Pharmaceuticals, as you mentioned, and I loved it. Really enjoyed working in this space and progressing my career. It's been a great run for 30 years.
No, it's exciting because I went to school up the road from you at Rutgers and also went into the commercial side. So it's exciting to, again, to have read through your journey and now to see you make the major hop into sort of the the area where our blood pressure gets a little more raised, and that's in sort of novel drug development. But I'm looking forward to discussing all that. Before we get into all that, I'd love to go into sort of the background story of NervGen, because there's several other names involved here. Dr. Harold Punnett, his daughter, Cody Darnell, and then also at least Dr. Jerry Silver, who serves on your board, renowned neuroscientist that's been spending a few decades in this space, sort of one of the major thought leaders of spinal cord regeneration. Talk a little bit of background here and how sort of these three forces came together to lead to the genesis of NervGen.
Sure, sure. And and they were very instrumental in my joining the company as well. I think my first discussion with anyone from management was with Harold. Dr. Punnett. Now, Harold's a scientist by training. He's a dentist, very successful businessman. Um, and in my first discussion with him, he told me about, how he founded the company. His daughter-in-law, Cody, as you mentioned, had a fall in 2016. She had, she fell and had a pretty bad accident and became paralyzed. And it was pretty devastating to him as he was telling me the story, explaining how, his son and his daughter-in-law's life changed forever and her journey, which led to his journey. He started to scour the internet and look for, who was working on spinal cord injury, nerve regeneration. And so between 2016 and 2018, when the company was founded, I think Harold looked at every single technology and science that was out there, and he came across Jerry's Nature paper, and that was kind of a turning point for him. He started having many discussions with Jerry, and then ultimately convinced Jerry and Case Western to outlicense the technology to him and a group of investors from Vancouver, who really in-licensed the technology and took the company public in a very short manner. So Jerry's, it was kind of a collision of Jerry working his entire career on nerve regeneration and different techniques to enable nerve regeneration, and Harold's tragic event of his family that came together that kind of really was the genesis of how this company came to be. And then I came many years later, but I had, my first discussion was with Harold, and the second discussion was with Jerry. And the passion that these two gentlemen had were fantastic. They both understood the technology a heck of a lot better than I had. They're both scientists. I'm a business guy. So it took me a little while to read a lot and have several conversations with them, as well as conversations with folks that I know, to work through what the technology was and what the potential of this technology was. And the things that drew me to the company were, this is fantastic technology, and if it translates into human data, the sky is the limit as far as, this is more of a platform than a specific drug.
Right.
And we could help so many different people. So I felt that, I could help this company get to the next stage of its life cycle.
Excellent. Excellent. Well, before we jump into a little bit of what the technology entails and ultimately how the drug development came off that, let's talk a little bit about some of these facts and figures. And you have these outlined in your corporate presentation on the website, and we'll link to all this. We're talking about something close to 20,000 spinal cord injuries every year. We have about 300,000 in the United States living with a spinal cord injury, and then about an equal amount more worldwide. And while the numbers obviously aren't huge compared to some of the other chronic degenerative diseases we talk about on the show, the financial numbers here get extremely large very quickly in terms of not just the rehabilitation cost and the hundreds of thousands, but the lifetime care into the millions. Walk us through a little of these numbers in terms of incidence, prevalence, and a little bit of how you look at this space, because it is financially a major burden as well as an unmet need.
Sure, absolutely. I gravitated towards the commercial first. That's the first thing I started to look at, or all the different indications that the company had outlined in their on their webpage. But started first with spinal cord injury because that's where the most preclinical data was. And I think that's where the nearest opportunity was. But like you said, there's 18,000 new cases a year, 300,000 people living with spinal cord injury in the United States each year. But the biggest numbers that I've seen when I came across all of the data were each case cost either the person or the system between $1 and $4 million over the course of their lifetime, which is a phenomenal amount of money, and it's a tremendous burden not only to the people who and the families who have to work through this situation, but also to the healthcare system because there's a lot of other events, urinary tract infections, hospitalizations. These folks need to get their houses changed around, and I don't believe any of those $1 to $4 million costs those are healthcare costs. That's not construction costs of building ramps into their house or changing the configurations of their homes. So the economic burden is phenomenal. It's greater than 50 million. I heard a one speaker, and I've been looking for this footnote ever since I heard the talk, and I still can't find it. But in Canada alone, I think it's the number one health burden of any disease state. I still need to validate that number, but I can't. I could certainly agree that it is a huge number. It's greater than 50 billion dollars overall. The impact, and that's if you take $1 to $4 million per person, multiply it by 300,000 people, it's a pretty massive impact to the system. So I think if we could help change that and give folks function back and lessen this burden, it's a great opportunity.
Absolutely. Absolutely. And the core again, and we'll get to the sort of the drugs in a little bit, but sort of the core concept here that Dr. Silver has been working on over the years has been focused on the scar tissue. And I know, so I guess most of us, when we think about scar tissue, okay, it's just laying down this fibrotic tissue, but no. Scars, as we know more and more about it, sort of in this era of all things genomics and so forth, are a really unique ecosystem, and the technology here ultimately focuses on two parts of that. Sort of one, and I'm speaking, okay, out of turn, two business guys talking here about science, but one, sort of this interesting set of molecules, these chondroitin sulfate proteoglycans or CSPGs, which are involved in sort of all sorts of cell adhesion, growth, and binding processes. And on the other side of that, what the CSPGs bind into, something known as protein tyrosine phosphatase sigma, which are sort of, in essence, what catches the scar tissue on the other side. So this process of synaptogenesis and the way everything sort of comes together in that little realm. I know I skewered that a lot, but I'll hand that back to you to
All
explain sort
right.
of why this is the focus of what's going on on
Sure.
the SCI front.
Sure. So I'll peel back a couple layers of the onion here, and bring up some of the notes that Jerry taught me. Jerry's a fantastic, very dynamic. If you get a chance to talk to him, or go online and just Google Jerry Silver, you'll see how passionate and dynamic he is. Not only is he a brilliant person, but he's a great teacher, and educator. So he educated me a lot. So did Harold on the technology. But I think one of the people who actually summed it up very quickly for me was actually John Ruffalo, who's on our board, who suffered a spinal cord injury. And John basically, and I'm stealing this line from him because he shared it with one of the investors that I was talking to, basically said, if you cut your hand or if you break your foot, your body repairs itself very well. Um, if you damage your spinal cord injury, or you damage your spinal cord, your body protects itself very well, and it inhibits repair, in fact.
Right.
So that's the kind of the first layer of the onion. If you dig a little bit deeper, what Jerry's work really discovered was Jerry discovered, like you said, CSPGs, chondroitin sulfate proteoglycans, and his initial work, he was wondering why nerves grow certain places and don't grow other places. And
Hmm.
and it's because of CSPGs. CSPGs inhibit axonal repair or regrowth or regeneration or remyelination, which is the protective covering around the outside of an axon. So there's several things that CSPGs do. They're very plentiful in the glial scar, as you mentioned, but their presence and their upregulation inhibit axonal repair, axonal regeneration, axonal remyelination, and plasticity in general, which is basically messaging going from your brain past the site of injury to your, uh, to the other side of the injury, and ultimately to your arms and your legs. So once he made that discovery of this inhibition or these inhibitors, he then turned towards, well, how do we stop these inhibitors?
Right.
And that's where PTP Sigma came in. They, through a collaboration with Harvard, Jerry discovered that PTP Sigma was the receptor that CSPGs were using to attach to cells. So, the PTP Sigma receptor is what CSPG is attached to.
Yep.
So now they started to work on, well, how do we disrupt this process? And Mark DePaul, who's actually on our staff, who used to be on Jerry's staff, and his team started to look at different products in different ways in order to disrupt this process and this upregulation of CSPGs. And we're actually still doing a lot of work today to elucidate the actual true molecular mechanism. But what we do know is that when NVG-291 is entered into the process, it does disrupt CSPG's activation, right? And it has been proven in many different animal models to not only remyelinate and regenerate and enhance plasticity, but those things will actually turn into function.
Mm-hmm.
So in vivo and in vitro, we have a lot of data that shows what happens in the tissue biologically when NVG-291 is put into the system or put into the process. But then we also have animal models that show, well, what does that mean?
Right.
It really actually turns into motor function. So there are two really key publications that we started to analyze quite a bit. One was done by Dr. Rink, the other one was done by Dr. Milton, and one was in the acute phase, which is shortly after the injury, and then one was in the chronic phase, which is after the body is repaired and plateaued. And we analyzed those studies because in the rodent models, they regain motor function, significant advancements in motor function. So, meaning you would crush the spine just like the biological process that happens in humans,
Mm-hmm.
and then give NVG 291. And rodents that couldn't walk started walking. They couldn't climb a ladder. They started climbing a ladder. So there was fantastic animal model data that shows that you could damage nerves and crush nerves, give NVG 291, and the animal would get back to almost normal behavior. So those two models, the acute model, meaning shortly after injury, and the chronic model, which is we call chronic basically after the injury, the body does repair itself for a period of time, but then it
Right.
plateaus. In animals, that's like 3 months. In humans, it's about a year. So the model after that recovery plateaued, we also gave the drug and saw that there was still improvements in those animals. So that's kind of why we set up our current 1B2A trial the way we did. But back to your question of how these things work, there's a ton of tissue data out there. There's a ton of animal model data. And right now we're doing a lot of genetic and molecular level data to see what are the specific
Mm-hmm.
signaling pathways that we're actually bringing back to life that have been,
Mm-hmm.
turned off from CSPGs.
Yeah, I think that's well, there's two really important things here, just for the audience. Uh, in addition to sort of enhancing the recovery, so you mentioned walking and climbing, also impacting things like bladder control, vision, some sort of memory issues as well. So that sort of points to in these studies not just the SCI indication, but the broad potential for this in the CNS. But another interesting thing I wanted to touch on a bit as well, because I think that we didn't get there yet. The NVG 291 is a rather elegant almost sort of a bifunctional compound in the sense that on one hand you got this intercellular sigma peptide, which is doing sort of a lot of the magic in terms of dealing with the protein tyrosine phosphatase sigma, but you created this shuttle as well that gets it properly into the nervous system. And so in this case, you're not injecting this peptide directly into the spinal cord. You're just putting it under the skin and it's nicely finding its way to the proper scar tissue. Could you say a couple words about that? Because I think this is another important piece of what you developed here, which is kind of elegant.
Yeah, so there's a lot of work actually being done right now
Yeah.
by Matvey Lukachev, who's our head of preclinical development in this area. Because I'll give you an example. In our stroke model, in the stroke studies that you elucidated, the drug actually worked distal and local. So there was local regeneration, but there was also some distal, meaning far away from the site of injury, repair as well. So we are actually in the process of looking at this from many different molecular and genetic angles to see what is the specific molecular mechanism of action. And this is through discovery. As you mentioned, it's a peptide. It's 35 amino acids, so it's a pretty large peptide, just short of a biologic, but it is injected daily subcutaneously into the stomach. So it is having a systemic effect. The question is how much of it is systemic versus how much of it is local, and we're
Mhm.
elucidating that as we speak.
Got it. Got it. Perfect. So you mentioned, obviously designing the phase one. You completed the phase one clinical trial in healthy volunteers. You have the 1b/2a, and again, I'd like you to talk a little because you mentioned obviously the subacute injuries, which are quite recent, and the chronic. And in chronic, obviously in humans here, I believe it means anywhere, which is quite exciting, from 1 to 10 years post injury. So, because I think normally you hear about, okay, the further we get out from an injury, the more difficult this becomes. But you're going to be recruiting quite a wide range of subjects here. Take a little time to walk through a little of what this study looks like, its design, and so forth. I think this is another important piece of the puzzle.
Sure, sure. As I mentioned, those two studies by Dr. Rink and Dr. Milton really kind of set the foundation for why we're looking at both what we call subacute and chronic, right? So chronic is in humans post 1 year. So what I mean by that is somebody has an injury, walk you through the kind of the patient pathway. They have an injury, they're hospitalized, and within the first week, they're in surgery to get to decompress the injury site to allow as much repair as possible. And then from that point forward, there's nothing for these patients. There's no drugs to enhance, axonal regeneration. And they are basically on their own to recreate connections themselves and push themselves to drive those connections. And there is a rehabilitation process that starts inpatient. So
Hmm.
the first 4 weeks are inpatient rehab. And then after that, they're discharged and they go into outpatient rehab. And during this process, they're still repairing themselves because you have an inflammation process that's dying down, and now you have the body's natural mechanisms giving them some function back. But then they plateau. Right around between 9 and 12 months, folks will plateau, and that's kind of where they are for the rest of their life. They've hit their peak level of repair at that point in time. So we wanted to study. Most drugs have studied acute,
Right.
meaning immediately after the injury. So it's a challenging place to study, not only logistically for trying to get somebody who's going through this traumatic experience physically and mentally to get them to sign up for a clinical trial.
Right.
Um, but their body's repairing. So the hurdle for the drug to prove that it works has to be pretty high. It has to prove that it actually works on top of the body repairing itself in that repair system. We're going to be studying it there. We started our studies post the plateau in the chronic period, mainly because we have good data that shows in the animal models there were plateaus, and then you treat and you could start to repair post that plateau. So if you can imagine, it's a lot easier to show if a drug has an effect in that stage. It's kind of an oxymoron. Most would think, well, you should treat early because you'll see the biggest response, and that may be true. But since we had the data in the chronic setting, we started in the chronic space because that's where, of the 300,000, 18,000 new cases a year, there's 280,000 people in that chronic space. That's a massive market. If we could show an effect in that space, not only is the bar lower for the drug to show an effect,
Right.
but the population is much greater.
Right.
So our studies currently enrolling in the chronic area. Once we complete with that, we'll go into the subacute. We define chronic as 1 to 10 years. We're redefining subacute to be 20 to 90 days because that's a key period after the surgery, after they've healed somewhat. They're still in the hospital, but they're making their decision to go to outpatient rehab. And that's where we'll get folks to sign up when they leave inpatient to go to outpatient. So the design of the study is patients have to have connectivity,
Okay.
right? So there's two types of injuries, motor complete and motor incomplete.
Yeah.
Motor incomplete is they have some residual connectivity, right? So we are making sure that folks have some connectivity, and we're measuring that through what's called electrophysiology. So they actually either through a wand or through electrodes put to the base of the brain, they'll send a signal to a sensor on your hand or on your leg,
Okay.
and they'll see, is there a signal, right? If there's a signal, even though the patient may be paralyzed, if there's a signal, that means there's some connectivity,
Mm-hmm.
right? So that's the primary entry criteria into the study. Um, and then we're measuring the improvement of that signal. So we're measuring the primary endpoint is electrophysiology. We're measuring does NVG-291 improve the strength of that signal, meaning are there more connections being made? The second thing we're measuring is the speed of that signal. So if the signal is faster, not only are we making more connections, but we're likely remyelinating broken connections. So that's the primary endpoint of the study. All of the secondary endpoints of the study are functional. They're motor functions. So it's 10-minute walk time, 9-hole peg test. These are
Yeah.
standard functional locomotive measurements that if you actually make connections and you prove that you've made connections, now you're proving that those connections are turning into action and
Mm-hmm.
motor connectivity and function. So we have a primary endpoint of connectivity, all secondary endpoints. I think there's 6 clinical secondary endpoints that are all function related.
Mm-hmm.
So this clinical trial is really designed to show do you make connections and show do you improve those connections turn into function so that we can tee up a next clinical trial, which would pick a functional endpoint based on everything that we've learned in this phase 1b/2a clinical trial. So the goal is have data in chronic, and we're expecting that early next year. And then we're moving into an end of phase 2 meeting with the FDA. We're proposing a phase 3 study, and we're off to the races.
Awesome. And when you mentioned the before when you talked about sort of the typical things that happen following these injuries in terms of the decompression and then the physical rehabilitation. How much of the sort of the physical rehab side of this is incorporated sort of as part of the studies, or I'm asking this in the perspective that when we talked about this theme before, there's always this thing comes up that if somebody hasn't walked for 10 years or has been incontinent or he's not sexually active, there's a lot of sort of quality of life issues associated with
Sure.
this disease as well. Sort of the follow up, because it's not, it could be right. You give someone this and they start get up out of a wheelchair after 10 years, but there's a
Yeah,
lot of things that have to happen in that process as well in terms of learning everything obviously below the injury again, that they might have not experienced or for years. Talk a little bit about that as well. I think that's
sure.
maybe a part of this whole theme that we maybe don't hear as much about as we should.
No, it's critical. We believe exercise, targeted exercise, is critical, and that's so. Dan Michael, our chief medical officer, has won awards for the design of this study. And we chose Shirley Ryan Ability Labs in Chicago as the single center for this study. So it's a challenge, right? Because these are folks that have recently had an injury, or in the chronic group between 1 and 10 years ago, they've had an injury. Now they have to move to Chicago for a 16-week study because it's a single center study. The reason we chose Shirley Ryan, there's two main reasons we chose Shirley Ryan. Number one, Monica Perez, who's the PI, is an expert in electrophysiology, what I
Mhm.
explained earlier. She's an expert in measuring the connectivity and whether the connectivity is going to improve. But number two, Shirley Ryan Ability Labs is the number one rehab hospital in the United States and potentially the world for the last 30 years. So part of our protocol actually incorporates an exercise routine that targets the muscles that we're actually going to be measuring the connectivity of. So you can imagine these patients, they're entered into the trial, they're randomized, drug or placebo. All of them go through this targeted exercise. So some of them are getting the drug on a daily basis for 12 weeks, and they have to go into Shirley Ryan Ability Labs, which is a very top-notch rehab facility,
Mhm.
and they're doing targeted exercise on the muscles that we're measuring function on overall. So I think that we've set this up so that we've stacked the deck essentially in favor of, if this drug works, we're going to see it because the patient's getting the drug, they're measuring whether it's connective through electrophysiology, they're going through exercise 5 days a week on the targeted hand and leg muscle groups. And then we're measuring whether those hand and muscle leg groups turn into mobility or function
Got it.
over time. So it's a critical component. Exercise is a critical component because this is, spinal cord injuries is a challenging, difficult space.
Right.
And you can regrow nerves, but if you don't work those connections, you're just regrowing nerves. You actually have to
Mhm.
turn those connections into life, into motion.
Yep. Yeah. I appreciate you bringing that up, and I just wanted to sort of for the audience to understand that for groups like you that are working about this, even though we want to think about a magic shot or pill, there's a lot that goes into sort of the ecosystem of this disease. And I'm,
Yeah.
I'm glad and again, I looked at the clinicaltrials.gov, and you can see sort of the really sophistication behind the sort of the primary and the secondary endpoints here and what you're looking at. So I think that's really cool. So, we talk about NVG 291 in terms of SCI, and obviously some of what you saw in the animal models in terms of applications for things like stroke. But it is your most advanced program, but you got other things in the pipeline. And I was wondering if you could say just a couple words. I know it's earlier stage, but you have NVG 300, which also, and I think, well, 291, you said sort of fell short of a biologic. This one is an actual biologic. Stroke, ALS, Alzheimer's, MS, spinal cord injury as well. So this one must have some, I see, interesting mechanisms. Whatever you can say about it at this stage, I know it's earlier on, but again, if you could take us into what you are excited about in terms of this candidate and why you chose it as part of the portfolio, that'd be awesome.
Yeah, we're not saying a ton about this,
That's
obviously,
cool.
because it's still early stage. We're walking the fine balance with whetting people's appetites from an investor perspective so that
That's
they know
cool.
we have something, but also validating it first. And that's where we are today. You mentioned early on, MVG 300 currently is in preclinical studies in animal models for ALS, stroke, and a confirmatory study in spinal cord injury. So we did one study in a more challenging rodent spinal cord model with a different type of injury, different type of rodent that is pretty challenging, and it did really well. So that's why we're doing a confirmatory study in spinal cord injury with NVG 300. And we've picked stroke and ALS as the two indications that we also want to get into. So we'll know in Q1 of next year if we have a drug candidate that we want to bring into IND enabling studies. We're very excited about this. This was an internal discovery made by Matvey Lukachev and through his work on MVG 291. And we believe that if we can actually bring this product forward as a product candidate, it gives us a lot of opportunity because a lot of big pharma companies are this is a molecule that it can get into MS, AD, ALS, similar to MVG-291, but it helps us split the program up a little bit.
Yeah.
And those types of programs are much bigger, much more expensive, and allows us to continue to focus on spinal cord injury, but have optionality to do further development programs with potentially other partners.
Excellent. Excellent. Yeah, again, I wanted to point out that yeah, you have more than one thing in a pipeline. And again, as this is a such a unique sort of biologic area, such a unique area in terms of the genomics and all the biology in terms of neuroplasticity and regeneration and remyelination and everything else that's happening in this biology and it's nice to see you got other candidates in there as well, thinking about it from all angles. So again, it's pretty cool. I know we can't talk much more about it, but maybe we'll do a follow-up episode
Sure.
when it gets further on down the line. Um, what else is happening, Mike? Anything else we should know about that's public facing? I know obviously you're doing the one study now. What's hot in terms of the rest of 2024 as we get into 2025 that you can mention? And then any other public facing stuff in terms of conferences, talks, investor conferences that you want to mention while we have here. Anything else that I missed, please?
Yeah, so we have several conferences that we're obviously coming up to from a financial perspective. I believe Stifel is our next one up in New York.
Hmm.
We have CEM meetings for some high net worth individuals down in Florida in November. We're starting to tee up all of our meetings for JP Morgan this year.
Right.
It's going to be a very busy Q4 and Q1 for the company. So our key milestones that are upcoming, obviously, everyone's anticipating when we're going to make the announcement that we've completed enrollment in the chronic cohort. And we are very close. We just put out a press release. We were targeting Q3. Obviously, we're at the tail end of October here. So we're obviously going to be in Q4 when we announce complete enrollment of a chronic cohort. And what that will translate into is we'll give folks exactly the date that we're going to have data. So it's a 16-week trial plus 4 weeks to gather the data, clean the data, lock the database, a couple of weeks for analysis. So 16 plus around 6 weeks, we'll have an announcement of
Nice.
data for the chronic cohort. We're then going to be transitioning, as soon as we complete enrollment in the chronic, we're transitioning into the subacute cohort, which is another 20 patients that were closer to the time of injury. And that will take place immediately after the chronic is enrolled. We also have, as I mentioned, data coming out in Q1 for MVG 300 and those three disease models: Alzheimer's, stroke, and ALS. So it's going to be a very busy time for the company. The next 3 to 6 months, we have a lot of data that's coming out for not only our pipeline, but for MVG 291 that's going to transform the organization. So what are we doing about it? Internally, we're doing a lot of planning. We're doing a lot of mapping out how we're going to transition this company from a TSX listed company to potentially uplisting on NASDAQ.
Hmm.
We're working with bankers as well as a lot of our current investors to talk about the timing of that. Do we do that in conjunction with the data readout of the chronic cohort?
Sure.
Because we don't want to lose any steps. We want to go immediately from data readout to finishing the analysis, submitting a package to the FDA, having an end of phase 2 meeting, and teeing up a phase 3. Obviously, we don't have the cash to do all that right now, so we're going to have to do a pretty significant raise, and likely that's going to be potentially in the U.S. as a NASDAQ listed company. So it's an extremely busy time for us right now. We're a small company, and we have to do a lot of hiring and bring a lot of talent into the organization at the same time. So we're building the plane, building the car, actually, I should say, and we're driving the car 60 miles an hour down the road at the same time.
Well, no, it's you're clearly making amazing strides. We chatted before the show. I love this space. It's clearly one that's as it's been unmet for so long that you obviously put together the right team, the right technologies, and I'm a big fan. I wish you and the best on both the financial and the clinical scientific front. And again, I hope we will be able to do a follow up next year when the 291 data comes in, or if there's new things to report on NVG 300. But really awesome work, Mike. Again, for everybody that's going to be tuning in across the various podcast networks or watching on our YouTube channel. Again, you've been listening to Mike Kelly, President and Chief Executive Officer, NervGen Pharma, doing really amazing things with the technologies to enable central nervous system repair on both the traumatic injury and the chronic disease front. Mike, again, I want to thank you for taking the time out of your schedule to walk through what you're up to. Thank you for everything you do for this patient population. And as we like to say on our show, thanks for helping to create a better tomorrow for so many people that are going to require your technologies and tools one day. Really an awesome story.
Thanks, Ara. Appreciate the opportunity. And, I'd like to thank all the folks that have entered into our clinical trials. They're helping the future and helping, the 300,000 patients out there waiting for something. So thank to them and their families and the sacrifice that they're making. But I appreciate your inviting us here today.
Great having you.